Here we go on to part 5 of the kidney. Finishing up. Okay, so I had briefly introduced that we're going to talk now about the effect of hormones on this system. So we've talked about the countercurrent multiplier system and how it makes it very salty in the kidney, and in that way we can reabsorb water in the nephron. Now we want to look at what happens in the distal convoluted tubule and in the collecting duct, because that's where the effect of these hormones is going to occur. So vasopressin, ADH, antidiuretic hormone. If it's antidiuretic, it means that it prevents the loss of water, prevents the loss of excessive water, and aldosterone. So let's look at those. Back to the nephron again, but the section we want to look at now is here. So the distal convoluted tubule and in the actual collecting duct, right? So it's highlighted in purple here. And it's telling you here in 3 that the permeability to water and solutes is regulated by hormones. So this is kind of, I think of it as the last chance to reabsorb water before it becomes urine. So you have to be able to retain some of that extra water, and then after that it's urine and it's excreted. So it's telling us in 4 that urine osmolarity depends on the reabsorption in the collecting duct, right? There's a classic question that always comes up during these lectures, and that is, you have the case of the girl pulled alive from the ruins. So the classic question of if you were trapped in a place with no water and you didn't know if they were going to find you or how soon they were going to find you, would you drink your urine? And what's the answer to that question? Because look here, it can be anywhere from 50 to 1,200 milliosmoles, which means it could be super concentrated or maybe not so much. And if it were not so much, it would sort of be okay to drink your urine if it was a life and death situation and it was going to save your life. Yeah. But if it's here, no, that would be worse. So if you were perfectly hydrated before you got trapped in this horrible place, usually the thought is you can do that once. Drink your urine once. After that, you start to become more dehydrated and it would just be very salty and you would be ingesting more salt, which would make it worse. So that's a good little tip. And now let's move on to the control of that sodium balance. So think about what happens when you eat a bunch of something very salty, like salty Fritos, right? You eat Fritos and you didn't realize you were eating that many. And then what's going to happen pretty soon is that it's going to trigger thirst, right? You're going to be very thirsty. And if you're not drinking, if you don't have something that's right there diluting all that salt, you're going to release vasopressin or ADH. And then what's going to happen? Well, you're going to be able to conserve water. And in conserving water, because you're trying to dilute all that salt, in conserving water, you're going to also increase blood pressure a tiny bit. But next, what's going to happen is that you need to rid your body of that extra salt. I mean, one thing is to just hang on to your water, but after that, okay, we need to excrete that salt along with that extra water. So all that control has to happen of that balance of when you eat too much of that salt. So ADH, antidiuretic hormone, or vasopressin, regulates urine production by regulating, now this is really important, regulating what? Regulating the permeability of the collecting duct to water. The permeability of the collecting duct to water. And it's going to be, there's 2 things I really want you to know, what it's doing and how it's doing it also, and why it's doing it. So this is going to be released in response to high blood osmotic pressure. So you've eaten your Fritos. That's essentially what it is. Remember it that way. So lots of salt, that's going to change the osmotic pressure. Change osmotic pressure is going to be detected in your body by pressure receptors, blood pressure receptors, and osmolarity receptors, and you're going to then release this hormone. You're going to release antidiuretic hormone. So in the drawing here, just to remind you, collecting duct is this end portion. This is where you're making urine, and you're going to want to be able to reabsorb more of that water. And by reabsorbing more of that water, what's going to happen is it's not just going to nowhere land, but it's going to come back into these capillaries that are in this region. So it's going to go back into circulation. Okay, so I have that return of that fluid not lost in the urine. And again, results from blood osmolarity. There are a couple of other mechanisms too, like blood pressure, but the most important is osmolarity. So I'm just indicating to you guys the most important of these. Okay, so how is it that it reabsorbs that water? So we need to talk about aquaporins. So look at that cell membrane again. And we have water channels, aquaporins, that are integral membrane proteins. And the individual, Peter Egri, got the Nobel Prize in Chemistry for his work on aquaporins. So who knew, right? We always said that water was a tiny molecule and it was able to sneak in through here because it was so small. And that's what I've been telling you so far, but I lied a little bit when it gets to this chapter, because actually there is a membrane protein, a channel here for water, and that is called an aquaporin. And these are exceedingly cool that you could have a channel that's going to allow water through. So this slide is very important. This is what I gave you for assignment for drawing this week. When you're drawing it, be sure to look at all the steps slowly so that you understand. And let's go first to what this is an enlargement of. What it is think of the vasa recta here, and this is the collecting duct, and it's showing the cells that make up the walls of that collecting duct. And what would be in the middle of it is the filtrate. So now we transpose to over here, and you'd have a little bit of the lumen of the collecting duct, which has filtrate, the cells that make up the wall of the collecting duct, and then just interstitial space a little bit, and then right bang up against the vasa recta that's going to take that water back. Okay? So you know where you are now. This is the wall that's holding this is the lumen of the duct. So this wall here is holding the filtrate. This is filtrate. And that's the filtrate is what we have to get water out of there. So what we want to do is get that water out of there and back into the blood flow, back into the vasa recta. But let's look at it in steps. So if we were to start here, we have vasopressin that would be circulating in the blood, correct? Because it's a hormone that you have released and now is circulating in the blood, and it needs to find a receptor that it's going to affect. So when it gets to this area of the kidney, step 1 here, it's going to bind to a membrane receptor on the cells of the collecting duct that are holding the filtrate. So here we are. And this is when we go back and remember those things that you looked at in chapter 3. So what happened is that when this receptor is on the outside, you had a step 2, which is a second messenger system, and the second messenger system is going to activate something that already exists and that doesn't need to be manufactured from scratch, correct? So it's quite quick. So step 3, what happens when that second messenger system is activated? It comes over to these guys. Look how cool these are. These are storage vesicles. These are aquaporin pores, little holes that are inside the cell. They're not doing anything there. But if you make them migrate and insert them here into the apical membrane. Apical means the one in the lumen, towards the lumen. So towards the apical membrane, and insert them here in that membrane, right? We just sort of insert them there and put in another one and put in another one. We have these vesicles migrating, and now we've created an opening for water to travel right into the cell. Oh, so cool. So here are these water molecules. They're coming in through these aquaporins, through just diffusion, because these cells are pretty concentrated, 600 milliosmoles. So they're high in solute. Water is going to be able to come in from that collecting duct lumen into these cells. But that's no good to just have them sit there. We want them to continue on their journey and come across over to the vasa recta that's quite concentrated also, because everything in the depths of the kidney are of a high salinity. So if this is high, water tends to dilute things, or solutes suck. It's going to bring this water out of the other end, the basolateral end of the cell, apical, basolateral, and bring it through osmosis back into the blood, back into the vasa recta. And we have just, in the collecting duct, reabsorbed some water. How did we do it? We did it with a hormone. The hormone has to first find its receptor, make those aquaporins, allow the water in, bring the water in, and drag it back into the vasa recta. So pretty cool stuff. So they are pretty brilliant, I think. Who knew that those were there? And for years we didn't know that they were there. And here are all the little water molecules then now have their own passageway through this aquaporin. Aquaporin's a good name. Aqua, water, pore, hole, or entryway. Aquaporin. Okay, that was all for vasopressin or ADH. Now let's look at the second one, aldosterone. What does that do? Aldosterone increases the active transport of sodium out of the distal tubules and collecting ducts. Wait a second, so it's not bringing water out? No, what it's doing, it's moving salt. Okay, so slightly different from ADH. Okay, it's transporting sodium out of the distal tubule, out of the nephron and the collecting duct. And by bringing salt out, I'll show you what the result of that is. I first wanted to tell you what is it released in response to, because before with ADH we were talking about osmolarity. High osmolarity, release ADH. Now, when do you release aldosterone? Well, you release aldosterone in response to low blood pressure. So think of this. If blood pressure were low and I could increase the volume of water in the blood, then that closed system of approximately 5 liters would have 5.5 liters, let's say, and it would increase the pressure in that system because of the increased volume. So it's going to react to that, to the low blood pressure, and in that case, aldosterone is released. So what's happening? It removes sodium from the collecting tubule, so here we are. It's going to extrude, move this sodium out. And it actually also brings some potassium in. It lets some excrete out. So we're talking always in this area of the collecting duct. This is high blood. This is an error, you guys. Low blood pressure results from low blood pressure, not high blood osmolarity. That was ADH. So low blood pressure. So the idea is that by just messing with the salt, I can make this region more salty and the filtrate less salty. Okay. Hang on to that. I want to tell you that most of the reabsorption of sodium and water is really early. So if we looked at proximal convoluted tubule, most of it happens in this region here. So even though we filtered out a lot of salt, we regain it back and we put it back into the blood. 15% of water is reabsorbed in the loop of Henle. So, 75 very early on. The next 15 are in the loop, and that leaves just that last, 15%, 10 to 15. And that's what aldosterone needs to remove that sodium from the filtrate. So we're saying here in this slide that it fine-tunes sodium concentration because 80 to 90% of it already was reabsorbed. So I want to say that kidneys really break the rules. Water doesn't just cross the membranes, it uses channels. Cells don't shrivel up in high salt content. They live, they're not destroyed. I want to back up a little bit though, you guys. I want to go and show you that if we make it more salty in this interstitial region, it's also going to allow this salt to come back into the blood, and then water is going to follow. So the way that aldosterone is helping that reabsorption of water is creating a concentration gradient for that water to leave that collecting duct. So we have kidneys breaking the rules, right? It wasn't till now that I told you about channels for water, and that before we always said if you put a cell in the wrong sodium concentration, sodium chloride concentration, is going to just, shrivel up and die, and they don't in the kidney. So that is a little confusing. And this was the last slide, I mean. You've made it through all the lectures in human physiology. Congratulations, you guys. Now to study all this, and thanks for listening. Lots of listening. Thank you again. Bye.